Today, I want to talk about whether fibroblasts can revolutionize cancer treatment. My name is Rachel, and I spent my summer in Dr. Chris Pinn's lab characterizing cancer-associated fibroblasts from pancreatic cancer patient samples. So a bit about pancreatic cancer. It has a really low 5-year survival rate of 8% and the third highest mortality rate of all cancers. And what's unique about pancreatic cancer is that it has a really low number of cancer cells, and it's actually mostly fibrosis. So you may know, but a majority of cancer research has focused on the cancer cells themselves, but what about the environment that surrounds them, or like this fibrosis in pancreatic cancer? So fibroblasts are the main or most common cell type found in fibrosis. And when they're found in a cancer environment, they're known as cancer-associated fibroblasts. So these CAFs, they have subtypes, one of them being myofibroblastic CAFs or MyCAFs, and these express a lot of alpha smooth muscle actin. So this summer, we isolated and cultured fibroblasts from patient samples, and then I used immunofluorescent staining and Western blotting to look at these cells. What's important about this is that hopefully sooner rather than later, we can use this process on these patient samples, collect information about their fibroblasts, and then give this information to their physicians to potentially help inform their diagnosis or therapy. And so the question I asked this summer was, do these CAF populations vary between patients? And if so, how? Well, my results would say yes, it does vary. So as you can see here, patient 8 versus patient 272. Patient 8 has a lot more red staining, which stands for alpha smooth muscle actin, which is highly expressed in the subtype MyCAFs. So here I would say that patient 8 has a lot more MyCAFs than patient 272. But the more important point of this talk is, why do CAFs matter? Why does all this research matter? Well, basically, fibrosis, or these fibroblasts, are thought to act in a way that protects cancer cells from treatment. And so understanding how these CAF populations can vary between patients could help us predict how a patient responds to treatment. And so we can also potentially target these CAFs to sensitize cancer cells to different therapies. And I mean, the meaning of my summer wasn't just how my research contributes to the scientific community, but also how it's affected me. I was one of the first people in my lab to work with cancer-associated fibroblasts from patients. And as excited as I was also extremely intimidated. So I dove into the literature, I learned more about it, and I've learned two things. So the first thing would be I've gained a lot of confidence in my ability to learn independently. And secondly, I've also learned to have humility. Just because an experiment doesn't yield the result that you expected, it doesn't mean the experiment was a failure. You could have just been wrong off the bat. And that just means you have to try again, rethink your hypothesis. And overall, it's taught me so much about pure perseverance. So big thank you to, if you'll let me switch slides, Dr. Chris Pinn, the Pinn Lab members, the CHRI, the USRI, and all of you guys for listening.